checked into my flight heading to Haneda Airport after receiving my ticket. The All Nippon Airways virtual assistant reminds me that there is a new biosecurity protocol being tested at John F. Kennedy Airport. After the 2029 H5N9 influenza bombing of Hangzhou Station that killed over 150,000 people in the Shanghai area, in the months after the attack and dealt over $100 billion in economic damage, global biosecurity measures were being fully rolled out. Before acquiring this ticket to Tokyo, I was notified that I had to get my DNA sequenced in order to travel outside of the country. A global biometric database was being constructed in order to mitigate spread of disease, viruses, and other pathogens. The database even prevented me from traveling to certain countries, because bacteria, mutations were evolving so fast in their country, it was dangerous for certain populations that have not developed strong enough antibodies to travel to other regions. No one anticipated how fast microorganisms would be able to mutate and outpace our technology, even with the advances in synthetic biology, 4D printing and rapid DNA sequencing. It was 2035, and we were successfully cloning extinct species and repopulating habitats. We were printing self-replicating organisms to combat pollution in waterways. We have never had so much control over biology, yet at the same time we were at its mercy. Viral mutations were happening so fast and so quick, mass-produced pharmaceutical drugs were no longer effective. It seemed as though there was a new disease outbreak every month somewhere in the world. I was not worried as the silk protein pill I had ingested hours before had already attached itself to my lower esophagus. The pill was programmed to infect my body 6 hours into the flight, a hyper-strain of Spanish influenza, just before the symptoms would start producing noticeable changes in my own body's microbiome. Let alone all my fellow passengers' microbiomes, I found myself inside an existential K-hole questioning how we as a species got to this point in history. First I printed my silk pills that had an inert host cells embedded in the silk proteins. I then printed the hyper-Spanish influenza strain appropriately called New World Flu. This was all encoded into this new strain to produce a much faster, deadlier, and infection flu strain. On the side, I really have to thank China's National Avian Influenza Reference Laboratory for developing over 125 strains of the H5N1 virus in 2010. This work was invaluable to developing a variety of hyper-deadly viral strains including trans-species viruses that have since been used in conflicts to target population food supplies as a soft weapon. The interesting part of this whole process was infecting the silk pills. The final move in this biotech process was just laying these silk pills in the flu culture, a low-tech finishing move. The proteins in the silk were designed to only allow the virus, only permitted release several hours after contact with human saliva enzymes. This in turn produced tiny little viral bombs that would explode with such density that the human host's microbiome is overwhelmed and attacks white blood cells that immediately go into overproduction of antibodies, accelerating the virus's spread exponentially. There's no turning back at this point. There are probably several others like me, traveling to different airports around the world. As I do not know the full details of this operation, the probable implications are that this is a first strike that would drain manpower and resources of state actors, dealing with a pandemic outbreak in multiple countries on multiple continents. Granting us a window that would allow us to digitally seize several city smart control panels.